is about template-free detection of macromolecular complexes. Our work basically belongs to the field of a new field of visual proteomics analysis. This field aims to systematically identify the composition and spatial distribution of macromolecules inside the cells, in their inside the single cells in their near living conditions. So the recent advance of the cryo electron tomography imaging technology make it is possible to for us to capture the 3D electron density information of the cells at their near living condition, which enables us to perform such a visual proteomics analysis. So here are some basic concepts of how the cryo electron tomography imaging processes. In such process, we first have a biological sample that is quickly frozen and put into electron microscope, and the sample is rotated at certain degrees, and then we let electron beam pass through this sample, and the projected electron density is captured by a CCD camera. Then, by rotating the samples in different rotation, we can collect a series of projection images, and then the projection images can be combined into a reconstructed 3D tomogram using the back projection reconstruction algorithm. So the existing visual proteomics analysis typically is done through a template-based detection process, where we have a library of templates of known structures, and for each template in this library, we can use this template to do a template matching against a given tomogram, and calculate the cross correlation by between the templates and the tomogram by exhaustively sampling all the rotations and translations of this template. And this computation is this process is a very intensive computation process. And by processing all such templates, we can obtain an atlas of the matched protein complexes in the cell that is captured by the tomogram. So although this template-based approach has been successful in finding the macromolecules of known structures, however, you can only find what you look for. So on the other hand, as we know, many complexes have unknown structures. And recent study shows that large protein complexes vary considerably in shape and composition across different organisms. In addition, protein complexes may exist in different conformational status. So therefore, to complement the existing template-based approach, so we have developed a template-free method. It has following several steps. First, given a tomogram, we perform image segmentation to obtain the segmented objects that may correspond to the macromolecules in the tomogram. Then we compute the rotation invariant image features and combine these features to determine the the similarity between the segmented objects and perform a clustering to obtain an initial classification of such objects. And then we further use a hidden Markov random field approach to perform a voxel level classification refinement. So for the segmentation step, we use a watershed algorithm to do the segmentation. Simply speaking, we first find the local maximas of the image of the tomogram, and then we lower down the image intensity cutoff and extend the regions starting from these local maximas until they met each other. And then we keep only the high intensity subregions to define segmented objects. So because the macromolecules, even if they are of the same type, they could still be oriented differently in a tomogram. So to characterize these macromolecules, we use rotation invariant image features. This is done at the voxel level. So first, for a voxel inside the region occupied by segmented object, we can define a shell that centered at this voxel and represent the voxel intensities on the shell using a function that is indexed in the spherical coordinate. Then we can have a set of concentric shells and its corresponding the voxel intensity functions. And for each of such a function, we can expand this function using a spherical harmonics expansion, and where A is the coefficient that are specific to different G function, and YLM is a basis function that has such a structure. And previous study shows that given such an expansion, the L2 norm of the inner sum is rotational invariant. Therefore, we can collect all the L2 norms from all these L's and all concentric shells to construct a rotational invariant feature vector for this given voxel. And we calculate the rotational invariant feature vectors for all voxels in all segmented objects, and perform a large-scale clustering of these voxels, of these vectors, to obtain a feature space neighborhood relationship that indicate which two feature vectors are similar. After that, we can combine this feature vector similarity to obtain a segmented object similarity between two objects, and using following simple equation where the numerator is the sum of number of feature vectors located in one object that have similar feature vectors in another object, and the denominator is the total number of feature vectors in individual objects. So after calculating this similarity, we can perform a clustering and obtain an initial classification of the segmented objects, and then we can assign a class label to each voxel inside of this segmented object and perform a further refinement. So to do this refinement, we use a Gaussian hidden Markov random field model, which consists of 2 random fields. It has an observable random field that corresponds to the image, corresponds to the voxel intensity, and a hidden random field that corresponds to the voxel class label that are to be inferred. So according to this model, the distribution of voxel intensity is conditional on the voxel's class label is follows a Gaussian distribution that has parameters that are specific to the voxel label. And And on the other hand, for each voxel, the labels distribution of each voxel depend on its neighbors. And in our implementation, we consider 2 types of neighborhood relationships. The first type is the standard neighborhood defined on the voxel grade of the tomogram, and the second neighborhood relationship is defined on the rotation invariant feature space obtained using the large-scale clustering. And we adapt existing popular iterative algorithm to perform this classification refinement. To test our method, we use the realistic simulated tomograms. Note that, remember, this is the tomography imaging process, and but in this process there are a large amount of different type of distortions. For example, due to the limit of the tilt angle range, we only have an incomplete sample of projection images, which lead to a missing wedge effect in the reconstructed tomograms. And in addition, such electron optical factors like contrast transfer function and modulation transfer function exist in the captured image, and also there are a large amount of noise that were coming from different noises and from different sources exist in the captured projection image. So here are some examples of our simulated tomograms. For example, in such tomograms, you can when the simulated tomogram is noise-free, you can observe the missing wedge effect and also the contrast transfer function effect that largely enhance the contrast between the boundaries of the macromolecules, and the large amount of noise plus other distortions make such a identification problem hard problem. So here is our randomized testing procedure. We first randomly put a number of different types of macromolecules into a density map, and to use this as a ground truth data. And then we perform a simulation to construct a simulated tomogram. And apply our identification method to such a distorted simulated tomograms and compare the identification result against the ground truth result. So here is an example of the performance that is averaged from 50 repeated, that is summarized from 50 repeated experiments. And from where we can see that the HRMRF refinement has improved the performance of the initial classification, and also when summarizing the classification result into the object-based classification, we can reach a performance that could be similar to the template-based detection method. And we have performed the test using other complexes other sets of complexes obtained from PDB, and we all get similar or even better results. In conclusion, so the template-free macromolecular detection is crucial for macromolecular structural inspection of cells in their near living conditions. However, the large amount of distortion inside the tomograms make such a detection a hard problem. Nevertheless, we have come out a proof of principle method that has been tested on realistically simulated data, and the result looks promising. And as for further future works, we focus on improve our method to handle the tomograms that are highly crowded, with highly crowded macromolecules. And I would like to thank my collaborators, Dr. Frank Alba and Martin Beck, and also thank Dr. Shihua Zhang for his good suggestions. And this work has been supported by following grants. And specifically, this presentation work was made possible by a travel fellowship awarded by ISCB with grant funds obtained from DOE, NSF, and NIH. Thank you.